Hello, my name is Laura and I'm going to be showing the Kras protein, both normal and mutated. So the Kras protein is kind of like a molecular switch that kind of helps control when cells grow and divide. And it sits on the inner surface of the cell membrane where it receives signals from outside the cell and then relays them to the cell's interior membrane, telling them when to grow, divide, or perform any other cellular functions. So the mutated version, Kras G12C, is the one that's more commonly seen in cancer, and I'm going to kind of go into depth of like how both normal and mutated kind of differ. So I'll be starting with the normal Kras protein. So kind of going based on what I said, like this right here is kind of like the globular structure, this flattened like clay part of it, and it represents the main body of the Kras protein. And so right here, there's like a little depression on like the Kras protein for the normal one, and it can be known as the GTP binding pocket, which I decided to like show here, and it's crucial because it's where GTP binds to the Kras. So when GTP is bounded, the Kras is active and sending signals, and then it's converted to GDP, the Kras then turns off. So this yellow ball right here with like the blue beads over it and like this black strip kind of represents GTP. So this black piece right here too is also known as the P loop, and it's important where like G12 is. It kind of shows the importance of like G12's position. So I also wanted to talk about the different building blocks of Kras, so like the primary, secondary, and tertiary structure. So the primary structure of Kras is its amino acid sequence, and it kind of shows like these beads right here on like the little clay beads on like the string. But I couldn't show all 188 amino acids in this model because that would take an insane amount of time. But I highlighted like the main critical like G12 position so you guys can have an idea, which is this light green bead right here in the normal model. And so for the secondary structure, these orange kind of like flattened spiral kind of shape clays are like coiled around, and they represent the alpha helices. And then these green pieces right here that are kind of like flattened rectangular, they are showing the beta sheets. So the secondary structures are the fundamental building blocks that fold together to create the overall like 3D shape of this normal Kras protein. And for the tertiary structure, well, that would just be like this whole entire globular structure that you see right here. And it shows how all the elements fold together to create a functional protein. So Kras doesn't really have a quaternary structure, but that's only because it works as a single unit rather than combining with other proteins to function. So I just wanted to point out a few limitations with this clay model, other than the fact that like this isn't obviously a real Kras model. But first off, real proteins, they're always consistently moving. These clay models, they don't move. So Kras actually shifts between different shapes as it functions. I can't necessarily demonstrate that here without physically constantly moving this clay model. And also the scale of these models are not true to size at all. I had to greatly exaggerate the size. Like if you look at it, it's almost like, I don't know, the size of the bottom of like this vase right here. The actual real Kras protein is not that big. I had to exaggerate the size just to like be able to show better the differences between the normal and mutated versions. But it's actually extremely small, like very, very small, atomic level kind of small. Also, the proteins, they don't work in isolation. So these models don't show how Kras interacts with the cell membrane or other proteins in the signaling pathways. It's just the Kras itself. And the colors also, like I just used the colors to kind of like distinguish like the differences, the different parts of these Kras models from the globular structure all the way down to like the internal stuff like the beta sheets. In the real Kras protein, they're not going to have these colors and everything. So the protein function happens through complex interactions between atoms, water molecules, and ions. And I'm not able to show that within the clay. But overall, I think like these clay models do a good job of showing kind of like how a tiny mutation in Kras can show such a drastic difference. And this understanding has led to the development of drugs specifically targeting the G12C mutation. And it can also like help and offer hope for patients with cancer like driven by this molecular change. So overall, when you look at these two Kras protein models side by side, you can kind of get a better understanding of the molecular shape, how it determines the function, and how a single change in the shape can have very profound consequences. And I think the whole entire point of the Kras protein and like showing the Kras mutated version, so G12C, excuse me, is just a powerful example of how understanding the molecular details of diseases can open the doors for like new treatments, and also giving us an ability to target the exact molecular mechanisms that are driving these illnesses, such as cancer, that we're focusing with the Kras protein. So yeah, I hope you guys enjoyed this video of me showing, the normal Kras protein. Oh, it just fell apart. But the normal Kras protein and also the mutated version of this Kras protein. So I hope this video helped a lot. If you liked the video, comment and like below, subscribe. Just kidding, this is not the start of my YouTube journey. But yeah, hope you guys were able to enjoy this video.